Morning, people. I hope you're doing well, my dear-lovelies, because what I've got, it may contain science. Every week, we share a chosen science because when I read it, it made me go, wait, what? There are lots of different flavors I've discovered over the last 3 years, too. Wait, what? There's, how could I possibly not have known that? Like, hiya, warm-blooded fishies. There's, oh, cool, how do they do that? Like, dissolving forever plastics with common chemistry items. But today is, hang on, yeah, I never thought about that, but how does that work? To wit, if people have to move around every few hours on an airplane so you don't get a blood clot, how come bears can hibernate all winter, never move, and never need to worry about that? Well, I never knew I needed to know this, but thank goodness science, out of the journal Science, is already on it. Okay, first off, why is the whole airplane deep vein thrombosis thing a thing? Well, when you sit still for hours on end in an excessively tiny seat, your blood flow is pretty slow. After all, you're not moving around much, your heart is not exactly pumping. And physics guarantees that blood flow is going to be slowest in the venous return from your legs. Veins are returning back up your legs, and they're fighting gravity to do it, especially with blood that has traveled from your heart all the way down your body and is trying to get back up. That's real far from the pump, so a lot of that pump's force is lost by the time the blood returns up your legs. When your blood flow is slow, you get increased inflammation, which really, it doesn't help matters. If our legs are congested traffic on the freeway and I add a bunch of emergency service vehicles taking up lane space, I have now made our slow traffic jam way worse. Now, imagine every silver car in this traffic jam is a platelet. Clot-forming fragments of cytoplasm that stick to each other and act as a slap patch to cover holes and prevent bleeding. Normally all the silver cars are zipping down the freeway, so they don't stick to one another all that much, but now they're all in this traffic jam together, trying to change lanes to get around emergency services, and all of a sudden, there's a lot of silver cars between here and my freeway exit, right? And there you are, the freeway traffic theory of deep vein thrombosis. Yes, I have achieved peak Los Angeles. So in principle, most of those same things should apply to hibernating bears, at least on a physics level. We know they're not running their heart rates super high and pumping blood super hard while they hibernate, so blood flow's still gonna be pretty slow, but, hang on, are we sure they don't have clots? Maybe it's just a risk bears are willing to take? Oh no, risk is for grad students. We know bears don't have higher risk of clots because the paper tells us they, quote, found no echocardiographic evidence of pulmonary embolism during hibernation, and found brown bears in winter had low D-dimer levels, a sign of clotting activation, compared to bears in summer. Of course, that means someone not only had to give hibernating bears an echocardiogram, but also had to get blood from both hibernating and active bears to make those comparisons. This study was a collaboration between cell biologists and wild animal specialists, though we can assume those wildlife specialists were the ones getting those measurements. We can't assume that, right? Okay, so they get all these materials for analysis. What are they looking for? Hibernation is associated with all sorts of changes in physiological signaling. I mean, metabolism's an obvious example. They get sleepy rather than snacky. So we know there's an opportunity here to change the rules of the road with respect to coagulation. But which rules of the road are they changing? Are they putting fewer silver cars on the road? Are they putting emergency services on furlough for the winter? Are they adding lanes to the freeway, which in this metaphor would be vasodilation? Remember, our blood vessels can expand and constrict to allow more or less flow as a means of maintaining homeostasis. And that is what this study is trying to figure out. We know tons of things change in a bear's physiology when they go down for nap time, but what specifically prevents clots? The short answer is yes. Biology is complex, and accounting for a whole bunch of anti-clotting genes were down, which should explain the overall lower D-dimer levels. But before I tell you the most prominent protein our researchers found, I need to remind you that scientists are irredeemable degenerates who do it for the memes. At the top of this paper, the editor has included a summarizing note with the title, "The Bare Necessities for Anticoagulation." If you think I am a pun lord, please know that scientists are my people and that this behavior is considered totally acceptable. That almost makes up for the answer to this study's question, which is heat shock protein 47. A tragically unfunny name. And maybe that's because heat shock protein 47 hasn't historically been a main character in vascular research. It is expressed on platelets, and its job is to be a so-called chaperone protein that helps other proteins fold, and in 47's case, that protein is collagen. Platelets activate when they see collagen. That's because usually all platelets see is blood vessel cells, called endothelial cells, as they travel to circulation. If you're in the circulation and you can see collagen, it's because that endothelial cell layer is missing, like a pothole in the road. So it makes sense that a cue for platelets to activate is when they call in emergency service, get sticky, all that, is when they see something missing. Heat shock protein 47 helps platelets tether to collagen and facilitates all that collagen-related signaling. So it makes sense that if platelets activate when they catch collagen, and you, a bear, have less of the protein that lets platelets catch collagen, you're going to see less platelet activation. Basically, without this protein, platelets are going to be a little more like see no evil. And by that logic, if we had platelets without heat shock protein 47, we'd expect to see less inflammation when an animal has restricted venous flow. After all, their platelets don't have that tether that tells them to activate, so we don't see the consequences of activation, calling in emergency services. And lo and behold, that's exactly what this study showed in a mouse model, where they replaced the mouse's bone marrow with one that had platelets that didn't have heat shock protein 47. That mouse experiment tells us it's not just that heat shock protein 47 happens to be the most downregulated of a bunch of different platelet-related genes, though it is. It's like 55-fold downregulated in hibernating bears, which is 10 times more than the next most downregulated protein at just 5-fold. This experiment also tells us what the brown bear's body is doing when that expression changes. There are 151 genes that change expression during bear hibernation in just platelets alone. So these knockout experiments are a lovely example of the methods biologists use to tease apart that monumental complexity. And for most papers, I suspect that would be where the paper stops. Like, we already have a really cool finding. We have this huge picture of like 150 changes to bear platelets when they go for a winter sleepy. And we've modeled the behavior of the most changed gene to see what it contributes to this phenomenon. We have a pretty good explanation for why hibernating bears don't clot. Well done, biologists. Well done, whatever wild child did an echocardiogram of a hibernating bear. That's easily enough to get you into a high-profile journal like Circulation, but this baby's in Science. They went harder. Because think about it. If you're a biped sitting in a plane, or a train, or a bus, or a car, for hours on end, it raises your clotting risk. But you know who's sitting? All day? Every day? Me! All the physics concerns about sluggish venous blood flow in your legs still apply to people in wheelchairs, sure. We sit more actively than y'all do sometimes, so we've got our hearts pumping more, but we're definitely sitting for like a road trip's worth of time often enough, and that's fairly immobile, so why aren't we at higher clotting risk? So now, researchers look at 2 human groups, bedridden and wheelchair users with spinal cord injury, as well as non-disabled volunteers who hopped on bed rest for about a month. Like, you ever see those get paid $1 zillion to lie in bed for a month studies? Is one of them. Lo and behold, the folks with spinal cord injuries have downregulation of heat shock protein 47 compared to bipedal active controls, and after about 27 days of bed rest, so do the non-disabled volunteers, along with a number of other platelet activation genes. The idea then is that if a biped is immobilized for hours at a time, there's an increased clotting risk. But if you're immobile or sitting for a long time, your body eventually adapts to readjust your clotting risk back to normal, and the mechanism that does that is consistent across mammalian species, from bears to mice to humans. Even though this paper goes above and beyond giving us the bear data and the mechanistic mouse experiments and the human translational studies, it still leaves a zillion questions to be answered, like which of the body's mechanosensory mechanisms sense that we're immobilized and tell platelets to calm down? When do they decide to kick in the downregulation? And why? Not to mention, what molecular signaling pathway do they use to do this? A bunch of our cells are weirdly good at sensing the physics around them. That's how you get phenomena like when the vibrations from the cat's purr stimulates bone healing, right? It's sensing physics. So now that we've identified a mechanism that keeps sleeping bears from getting clots, the next step is to work backward and figure out the whole pathway from not moving much to, hey, blood flow has actually kind of been slow like this for a while. I'm gonna let platelets know. Ease up on that heat shock protein 47. That makes this paper a really strong representation of what modern biological basic science research can actually do. And this thing's an absolute showcase of different experimental models and state-of-the-art methodology. This paper doesn't set out like, I'm gonna find new treatments for one particular clotting disorder. Instead, it starts from a weird observation, namely, that immobilization doesn't cause clotting in hibernating bears, and by following down the rabbit hole, or bear lair, as the case may be, we're going to learn a ton about the whole story of how clotting works. As we move forward down this line of inquiry and uncover that pathway, we're not just going to find things that might help one clotting disorder. This approach lets us find a ton of different potential spots in that path that could help a ton of different clotting disorders, depending on what in the pathway is going wrong in that particular condition. Basic science not only satisfies our curiosity, like now I know why bears aren't at high risk of clots and why I, as a wheeler, am not either. But when we do get to the applied science stage and start testing treatments, we have enough context to know which therapy will be like, hey, this one little hack fixes your clotting issue, versus, hey, this fixed your clotting issue, and caused like 20 other problems. I'm helping. For some people, the outcome is the only point of science. If we don't make something out of it, it's useless. But here, on May Contain Science, answering curiosity is its own reward. So those folks are free to let sleeping bears lie. We're busy giving them echocardiograms.